Welcome to the sixth and final video in this short series on stereochemistry. To review, stereochemistry, or the existence of stereochemistry, was first discovered when people noticed that certain molecules would bend light. It was then discovered that the molecules that bent light did so because they had chiral centers, carbon atoms with 4 different groups on them. That light could either go to the clockwise or counterclockwise. You could call it D or R if it went clockwise. You can call it L or S if it was counterclockwise. Either way, you got stereoisomers. Those stereoisomers could be enantiomers, or they could be diastereomers, or meso, or even epimers. All in all, the whole thing is really simple, right? Obviously not. Stereochemistry is one of the more difficult subjects to get a handle on in the field of chemistry because of all the possibilities. In this video, to try to make things slightly easy and give some perspective, it's a good idea now to go over the history of these names. Essentially, the confusion in naming these types of compounds arises from historical fact. Optical activity was first observed in 1811, and this was observed in known molecules with known physical and chemical properties. However, the systematic way of naming such molecules by IUPAC only came into play in 1919. So essentially, any naming confusion or controversy is simply a case of the cart of the discovery of optical activity in known molecules being placed before the horse of IUPAC. So, as mentioned, in 1811, people noticed that biomolecules would bend light either clockwise or counterclockwise. These molecules were basically divided into 2 known categories of well-studied things, starting with carbohydrates, essentially sugars, such as glucose, which could occur in the D or L, mannose, also D or L configuration, and allose in the D or L configuration. These molecules had different chemical and physical properties and were named as individual different molecules. However, if you look at IUPAC naming, all 3 of them, or actually all 6 possible isomers, are isomers of 2,3,4,5,6 pentahydroxyhexanal. They're essentially different forms of the same molecule. Similarly, optical activity was also discovered in what are called amino acids: alanine, glycine, valine, and leucine, for example. These, once again, were compounds whose properties were known, so they were given different names. But under IUPAC, they would all simply be some sort of 2-amino followed by some chain, something enoic acid. Again, different molecules in this case, but all bearing essentially the same name. So, what happened is what always happens when people get together: an argument. The people in IUPAC decided they had the best way possible of naming any molecules, and this was very systematic, and everybody should do it. Unfortunately, the people who had been studying them already were set in their ways, and to this day, absolutely refused to change the names. So therefore, you're stuck with 2 separate ways of naming molecules. Sugars can be glucose, mannose, or allose, even though they're all pentahydroxyhexanal. That name is generally not going to be used. Same with the amino acids. The common names predominate over the IUPAC names. So it would seem that one has 2 separate naming protocols to remember. However, if one looks at alanine, for example, one can see that this actually comes in 2 stereoisomers. Depending on the position of the NH2 group spatially, L-alanine can be written as 2S2-aminopropanoic acid. D-alanine on the other hand, is going to be 2R-aminopropanoic acid. You would think you now have 2 compounds with 4 names to remember. But the important thing to recall is that you are actually looking at biomolecules, things that exist in nature. And these are made by enzymes which in and of themselves have chiral centers and are essentially engineered to only go one way. So in the case of alanine, even though you have 2 possible compounds with 2 possible names each, the reality is over 99% of the alanine found in nature is going to be the L form, or the 2S amino acid. Similarly, with sugars, you look at 2,3,4,5,6 pentahydroxyhexanal, the basis of the 3 sugars we've already seen. In this, you'll see there are 4 chiral centers. Theoretically, there are 16 possible stereoisomers of 2,3,4,5,6 pentahydroxyhexanal, and these have been prepared in the lab. However, practically speaking, once again, these carbohydrates are biomolecules, and they're made by enzymes. So in nature, one winds up with the glucose, mannose, and allose as distinct molecules, where the other and only the D form of these molecules appear in nature. So out of the 16 isomers, actually only 3 are common in nature and won't be found anywhere outside of a lab. So in essence, the thing to remember when looking at stereosensors is that they are predominantly found in biomolecules such as enzymes, proteins, carbohydrates, amino acids. IUPAC, on one hand, is useful because it's systematic. If you know the system, you can name every single molecule. On the other hand, it gives you it allows you to see all of the possibilities for any given molecule. In the case of stereochemistry, this largely is not necessary, as only a few of the possible isomers are known in nature. The common names, on the other hand, have the downside of having to memorize them. On the other hand, if one is studying biology or biochemistry, those names do constitute everything that's known in the field, and each one of these molecules, even though they are isomers of the same molecule, actually perform separate and completely different functions in nature. I hope that helps clears things up, or at least gives you an idea as to how to approach stereochemistry and how to study biomolecules and stereoisomers. And thank you very much for listening, and I hope you found this series enjoyable and useful.